Hello again. So this section is all about factors affecting transpiration and translocation. So we've been looking at these three stages, and the last one we looked at was the movement through the leaves and water loss through transpiration. So now we're looking at factors which affect transpiration. So what makes water evaporate from the mesophyll and then travel out the stomata faster or slower? Now remember, transpiration is when water evaporates from the mesophyll cells through the airspace and then diffuses along a water potential gradient out into the atmosphere. So most of the factors which affect the rate of transpiration are something to do with the stomata. This diagram shows basically what different factors. So factors affect transpiration can be split up into external factors, which is basically due to the environment that the plant is in. So things that we're going to look at here is temperature, humidity, light intensity, air movements, and soil water availability. Internal factors are features of a plant which will affect how much transpiration takes place. So that can be things like how many stomata it has in a certain area, its surface area of its leaf, and how thick its cuticle is. But as I say, most factors have something to do with the stomata because that's where the water leaves the leaf entirely. Now I think we'll start off looking at internal factors. So firstly we have cuticle thickness. So basically plants can actually lose water through their cuticles or through the stomata. Obviously most of it goes through the stomata, but different plants will have different cuticles. Now remember, the main function of the waxy cuticle is to prevent water loss. So it makes sense that if you've got a thick cuticle you'll lose less water, and if you've got a thin cuticle you'll lose more water. Leaf surface area is also an internal factor. So obviously the larger the surface area, the more evaporation that takes place, and that's largely due because larger leaves will have more stomata. So if we took two leaves from the same species, one was this size and one was this size, obviously this one would have a greater surface area, and so because of that it's going to have more stomata than this one, which would be smaller and have fewer stomata. So this one would lose less water, whereas this one would lose more water. So larger surface area equals more stomata, so there's more transpiration. And the final internal factor then is stomatal density. Now some of you might get confused with the density that we're talking about, especially if you do physics. You'll think it's something to do with mass and volume, but it's actually not in this case. Stomatal density refers to the number of stomata in an area of a leaf. So say for example you measure the stomata in 1 centimetre squared of it and calculated how many stomata there was, that would give you the stomatal density per centimetre squared. Now the higher the density, the more stomata, so the more evaporation will take place. So if you compare these two squares, this one has got fewer stomata in this area than this one, which has more stomata, so it's going to lose more water through transpiration. And that's basically just because there's more pores for water to leave the leaf. And that's the internal factors then. So stomatal density, leaf surface area, and cuticle thickness. The external factors then are temperature, humidity, light intensity, air movements, and water availability. Now the external factors are all to do with the environment that the plant is in. So the first one's light intensity. Now light intensity doesn't directly affect the rate of transpiration, so it has its effects indirectly. And this is why. During the day when there is light, the stomata will be open. This allows gases to enter the leaf so that photosynthesis can take place, but it also allows the evaporation of water. During the night, most plants will close their stomata because there's no light, so they won't be photosynthesizing, so there'll be less water lost. So light intensity indirectly affects it. When there is a high light intensity, the stomata are going to probably be open, and if they're open it makes sense that there would be more transpiration or more water loss. Whereas at low light intensity, the stomata are likely to be closed, so there'll be less transpiration. The second external factor we'll look at then is temperature. Now, there's two leaves here, but in general, if there's higher temperatures, there should be more transpiration taking place. Now, here's why. So this leaf is in high temperature, and this one's in low temperature. Now, this just shows the water molecules within the leaf on the left, and this shows the water molecules in the leaf on the right. Now, at high temperatures, there's basically faster rates of evaporation, which is just like in normal life. So that means that there's going to be more transpiration, or a faster rate of transpiration. Whereas at low temperatures, there's going to be a slower rate of evaporation and therefore a slower rate of transpiration. Now remember, as well as that, higher temperatures increase the rate of diffusion. So we should say here that at higher temperatures, there's faster rate of evaporation from the mesophyll cells and a faster rate of diffusion out through the stomata, so there's more transpiration. Now, the next external factor is humidity. So humidity is basically how much water vapour is in the air already. So this diagram shows a leaf in an area of high humidity. So there's a lot of water vapour around it. This means that there's a high water potential both inside the leaf and outside it. This means that we have a water potential gradient which is too shallow. So therefore, the water vapour builds up outside the leaf and little evaporation takes place. And that's simply because the water potential gradient is too shallow, there's not much diffusion taking place, so there's a low rate of transpiration. This diagram then just shows us what happens if there is low levels of humidity. So there's the water molecules inside the leaf and the water molecules outside the leaf. So within the leaf there is a high water potential, and outside the leaf there is a low water potential. So we've got a water potential gradient from the inside of the leaf to the atmosphere around it. So therefore we've got a steep water potential gradient. This means that more evaporation can take place, and so there's a higher rate of transpiration. So to summarise that, if there is low humidity, there should be steeper water potential gradient, and therefore more transpiration. So the next external factor is wind speed. So here we've got two leaves again, showing the water molecules inside. Now this one's in still conditions. And what you can see here is the water molecules diffuse from inside the leaf to outside, but then they stay there. Whereas in windy conditions, the water molecules diffuse out of the leaf, but then the wind comes along and carries them away. So you have got a really steep water potential gradient from inside the leaf to outside. And because that wind carries away all the water molecules from there, it maintains a steep water potential gradient. So in still conditions, as we say here, there's a buildup of water outside the leaf, and that's known as a diffusion shell. So that diffusion shell is basically all the water molecules which have built up there. So we've created a shallow water potential gradient there. So that means that there's less evaporation which takes place, and as a result, less transpiration. But in the windy conditions, the wind carries away the diffusion shells, so there's a steep water potential gradient from the inside to the outside of the leaf. So more evaporation takes place, and as a result there's more transpiration which takes place. The final external factor then is soil water availability. So this is basically how much water there is available in the soil. So during hot, dry summers, there may be less water available in the soil caused by reduced rainfall. There will also be less water available to plants. So what plants will do is close their stomata to prevent further water loss, which reduces the rate of transpiration. So if there's not much water available in the soil, the rate of transpiration will be lower. So to summarise all this then, if there are high temperatures, there's a high rate of transpiration. If there's high humidity, there is a low rate of transpiration. High light intensity indirectly creates a higher rate of transpiration. More windy it is, the more transpiration there's going to be. The more soil water availability, the more transpiration there will be. The more stomata there will be, the more transpiration will take place. The larger the leaf surface area, the more transpiration. And finally then, the thicker the cuticle, the less cuticular transpiration is going to take place. Okay, so the next section in this booklet then is translocation. Now translocation is another mass flow movement, but this time it's transporting sugar in the form of sucrose. So basically what happens is, if this is a little plant, there's its roots, there's the little leaf, sun shines down on the plant, it photosynthesizes, it produces glucose, it converts those to sucrose and transports them through the phloem to different parts of the plant. It might transport them down to the roots to store them, it might transport them to fruit to store them, or it might transport them to the growing regions to grow more new leaves and new structures and things like that. But either way, translocation is the movement of sugars, and it's the movement of sugars through the phloem tissue. Now, you don't actually need to know much about the process of translocation, but I think it's important to understand what's actually happening so that we can understand this little bit about the evidence of translocation. So sugar is manufactured in the leaves by photosynthesis and is transported primarily as sucrose to other parts of the plant where it is used in growth, development and repair, or to and from sites where it's stored as starch. Amino acids are also moved from cells where they are synthesized to places in the plant where proteins are being synthesized. Sugars and amino acids are transported in the phloem tissue, and this movement of soluble products from photosynthesis is called translocation. There are two key features associated with translocation. The process is energy requiring, and a two-way transport exists, i.e. translocation can move sucrose both up and down the sieve tubes, which you'll remember were the main structure in the phloem. The evidence for translocation being enacted, well, in fact, you know what, before we get on to this, let's look at translocation. So, as I say, translocation involves moving substances as a mass flow movement from source to sink. So let's say that light shines down on the leaf cell, which is the source. It creates these sugars in photosynthesis, which is mostly glucose, but then it gets converted to sucrose so that it can move it. The leaf cell is going to transfer the sucrose to the companion cell beside the phloem, and the companion cell is going to use energy, use ATP from aerobic respiration to pump the sucrose in here. So we basically have a buildup of sucrose in there. Now what happens is, because we've put all this sucrose in here, the water potential of this area becomes more negative, or we can say that there's a lower water potential. Because we've created this area with a lower water potential, it draws water from the xylem. So the xylem moves from an area of higher water potential to an area of low water potential. When the water moves, it actually creates a solution here, which is pushed downwards to, in this case, the root cell, which is the sink where it's going to be stored. And then we've got this sucrose, which is moved down, which then moves to the companion cell, to the root cell, where it gets stored as starch. Now, because we've just got rid of that sucrose there, we now have a higher water potential here, and the water molecules move back there, and they move up through the plant because of the transpirational pull and the cohesion tension theory. So that was translocation. It basically involves the movement of sucrose to the companion cells to the phloem, and then the movement of the sucrose down the phloem to where it's going to be stored. Now, the point of translocation is to move sugar so that it can be stored, or so that it can be moved for growth and repair. Now, as I say, under the current spec, you don't actually need to know the details of translocation, but what you need to know is the sugar that's moved by translocation, which is sucrose, and also evidence that it is an active process, which means what's the evidence that it actually needs energy in the form of ATP from respiration. So here's the evidence that it's an active process. The rate of flow is higher than can be accounted for by diffusion. So diffusion is generally quite slow, and because this involves moving sucrose over large distances relatively quickly, diffusion just wouldn't account for that rate of movement. As well as that, we talked about this earlier, companion cells have a particularly high density of mitochondria and are more metabolically active than other plant cells. And that's because they create ATP, which is used to pump sucrose into companion cells from where it enters the sieve tube elements via plasmodesmata. So the companion cells contain lots of mitochondria, they create lots of ATP, and they pump the sucrose from the companion cells into the sieve tube elements. As well as that, metabolic poisons, for example potassium cyanide, stop respiration, so they also stop translocation. So basically, if there's no respiration taking place, there's no translocation. Now, I'm going to skip down here a bit. How they can actually tell whether or not translocation is taking place is by using radioactively labelled sucrose. So this is sucrose produced using carbon dioxide with the carbon-14 isotope, which shows that sucrose can move both up and down the stem. So this is really clever what they do. They basically take a leaf here, and what they can do is, instead of using normal carbon dioxide, which uses the carbon-12 isotope, they use the carbon-14 isotope, which is radioactive and will show up under X-rays. So what we can actually see here is, this is a little leaf. Now, what they would have done in this case is they would have actually put a little bag around this. And within that bag, they would have put the radioactive carbon dioxide, put it into that bag, allowed this leaf to photosynthesize, and then they've put it under an X-ray after a certain period of time to see where all the sucrose has been transported to. Now, you can see here that lots of the sucrose has been transferred into this leaf, which is quite close to it, but also a lot of it has gone down the stem, which is probably likely to mean that the sucrose has been transported downwards so that it can be stored. Now, as I say then, translocation occurs, or the movement in translocation occurs from source to sink. The source is the organ where sugar is produced in photosynthesis or by the breakdown of starch, for example the leaves, and the sink is the organ that consumes or stores the carbohydrate. So the sugar can be moved to a developing bud at the shoot tip or down to the roots. But as we explained earlier, source and sink depend on the season. A potato plant is a sink during the summer as it builds up stores of carbohydrate, but it is a source in spring when starch is broken down to supply the energy for growing shoots. And that's everything you need to know about translocation.